Welcome everybody, and thank you for joining to our second webinar. For those of you who didn't have the chance to join us in the first webinar, I will shortly introduce myself and the GDRF activities. My name is Gilad Bennett, and I am the father of Adam, an amazing boy, three years old, who was diagnosed with GRIN 2D mutation, the rarest among the GRINs, as you know. I'm one of the board members of the GDRF. Before I continue, I'll just introduce the agenda. First, we will have a session with Dr. Tali Garin about gene therapy of GRIN and how it can help our children. Afterward, we have a short session, intermediate in the middle. And the last, we will have a Q&A section where you will be able to ask any question, and Tali will answer all of them. So before we continue to Tali's section about the gene therapy, I wanted to explain a little bit about the GDRF. The GDRF is a non-profit organization with the sole purpose of finding the cure for the GRIN. All the GRINs, it's important to mention, we are researching all the GRINs without any differentiate between the GRINs. And we differentiate only between loss of function and gain of function. So it's very important. This is the only differentiate, but we research gain of function and loss of function as well. The GDRF Foundation was established just 6 months ago, and already has numerous active researchers that research the subject in academic laboratories who decided to take this project on, and they are working on a full-time basis. So we have several researchers from distinguished academic laboratories that research, doing active research on finding a cure to GRIN. Each laboratory is specialized in different field of research, and all led by lead researchers in their field. One example is Professor Danny Offen from the Tel Aviv University, who led several scientific breakthroughs in ALS and other diseases. In order to find the cure we hope for, we need to join forces, all of us together, all the parents, all the different foundations worldwide, for the sake of our children. It's very important that we all come together and work for the single goal. All of you that can get involved and become a member at the GDRF, we welcome you all, and get constant updates on the research progress and influence the process. You can join free to be a membership as part of the GDRF, and we welcome all of you. And without further ado, let me welcome Dr. Tali Garin, who will discuss gene therapy and how it can cure our children. So please, Tali, you have the mic, and we will continue the discussion afterwards.
Hi, everyone. Thank you for joining us tonight. I would like to start by reminding you about the first webinar that I was talking about the several genes that are involved in GRIN disorders, and information about the protein, the NMDA receptor, which is critical for learning and memory, and is placed in the brain, making connections between nerve cells. And then I also discussed several tools that we have for studying the effect of the GRIN genes and the mutations in GRIN genes, namely the mice and the personalized model of creating nerve cells from every child with GRIN disorder. Okay, so GRIN disorders is a group of diseases that can stem from several genes that are involved, namely GRIN1, GRIN2A, GRIN2B, GRIN2C, and GRIN2D. It takes only one gene and only one copy of each gene to cause a disease. There are several ways how these genes be mutated. The first and the more frequent is a missense mutation. It means that one base of the gene is switched to a different one, and this causes an abnormal protein. The second type of mutation is a nonsense. In this case, the protein is truncated. It doesn't lead to the full length of the protein, and in nonsense and in deletion, there is less of a protein. So in these two situations, even though we have two genes, it's enough to have a mutation in one gene, and it causes a disease. So missense mutations are the most common cause of GRIN disorders. What happens in this situation is when there's an abnormal protein, not only does it interfere with the NMDA receptor, but it also takes over the normal copy. This is called the dominant negative effect. This is why, even though only one copy is mutated, we have a very severe disease. How can we approach this situation in gene therapy? So we can think about gene editing therapies, and I will discuss it later. The other type of mutation is a nonsense or deletion. Deletion meaning the part of the gene or the entire gene is missing. In this situation, there is only one functional copy of the gene, and therefore there is less protein that is produced. There is less NMDA receptor. So in this type of mutation, there is no dominant negative effect, but there is half the amount of the protein, and this is why it's usually a milder disease. But still, this is a very significant disease. How can we approach this situation? So there are two options. One, we can add a normal copy of the GRIN gene. It is called gene replacement therapy, but there's other alternatives that I will discuss later. So gene therapy is a very wide subject. Is it a fact? Is it a fiction? Definitely not a fiction. This is something that is turning the medical world upside down. I would like to mention a recent drug that entered the market. It is an FDA-approved drug therapy for a disease called spinal muscular atrophy, SMA in short. This treatment is a one-time treatment given to this disease that is causing muscle wasting and actually mortality of infants. It is not common. It is 1 to 10,000 births. And what's so amazing about this treatment is that it addresses the genetic root cause of the disease. If in this disease, which is not as GRIN disorder, when one copy is mutated, here there are two copies that are mutated, and there's lack of a protein called SMN. And for this reason, when there are no functional copies, the option is to give a normal gene that codes for the SMN protein, and it is done by a single intravenous infusion. So a little bit about SMA. It is very rare. It is 1 to 10,000 children, and it causes a death of nerve cells that are responsible for motor activities. At the end, it causes a paralysis of muscles and inability to breathe by your own. It starts at the age of 6 months, and it's one of the leading genetic causes of infant mortality. So in this disease, there is a genetic defect in the SMN gene in its two copies. There's not enough protein to make the nerve cells alive, and they will die. Eventually, by the age of 2 years, it's either the children need permanent ventilation or they don't survive. But this breaking discovery of the gene therapy allows these small children to receive one single shot of the SMN gene. And by that, by the age of 2, none of them required permanent ventilation, as opposed to only 8% from the natural history of the disease. The treatment was developed by Avexis and was purchased by Novartis. This is a very expensive treatment. It costs $2.1 million. It's actually the world's most expensive drug. And the reason is that it's based on a lot of years of research, of clinical trials. But I'm sure that eventually, when there are more and more gene therapy treatments, the price will be much lower. Now, Avexis has more programs entering clinical trials in the following years, also including Rett syndrome and ALS. And I'm quoting for the website, "We are at the forefront of an exciting time in healthcare when we'll be able to see major advancement in medical care with potentially curative gene therapies." How does gene therapy works? So it takes advantage of an adeno-associated virus. It's not a real virus. It cannot replicate or cause a disease. And inside its envelope, the DNA that we want, the normal gene, is inserted and delivers inside the patient's own cells. It doesn't change the DNA of the patient. The advantage of the AAV9 is that it can enter the brain and it doesn't need to be given directly, so it can be given by an IV infusion. And this is a one-time changing life event. So here is the scheme, how it works. So the AAV carries inside of it the desired genes. It binds to the cell membrane and then injects the DNA into the cell. The vector is, the AAV breaks down. It's not active anymore, but the gene is stable and it enters the cell nucleus. There it will start producing the required protein, the SMN protein, that will keep the nerve cells alive. There's a nice cartoon that you can watch. We don't have time for it right now. Very interesting about how gene replacement therapy works. But gene therapy is not only for rare and fatal diseases. This is another example of a gene therapy treatment for inherited vision loss. Manufactured by Spark Therapeutics, also very recently entered the market, also very expensive. Once again, it's an AAV vector-based gene therapy that is given by a one-time shot and replaces the mutated gene, in this case, that causes retinal dystrophy and blindness. Not yet FDA approved, but definitely in clinical trials, is the example of gene therapy for another disease called AADC that has many similarities with GRIN disorders and also was posted in the GRIN research discussion website. Very interesting. In this disease, AADC deficiency, the children are experiencing severe and global developmental delays with weak muscle tone, hypotonia, involuntary movements, and epilepsy, and require lifelong care. It is very, very rare, less than 1 million patients, and there are no approved treatments for this disease, same as for GRIN. Now, there are several clinical trials recruiting patients that in a one single technology of gene therapy rescue the amount of the protein in the brain of these patients and gives hope for more rare genetic neurological disease. the improvement of the children was remarkable as they started walking and communicating and really changed the way they function in society. So it gives a lot of hope for the same here in GRIN disorders. Until now, I talked about gene replacement therapies. When there's a gene that doesn't function, that doesn't produce the right protein, and we add something that replaces it. But there's another path called gene editing, also very promising in a way to treat genetic diseases. And I will discuss CRISPR, modification of CRISPR called dead CRISPR, and also base editing. These are very new tools that we have now in genetics that we didn't have a few years ago, and change the way genetics can not only diagnose diseases, but also cure them. So what is CRISPR? What is the big fuss? CRISPR is actually a molecular scissors. It can cut DNA. Before CRISPR, there were alternatives that were too expensive, too cumbersome, were difficult to manipulate, and CRISPR really made a revolution in the field. CRISPR is made up of two parts. The first one, called Cas, it is the actual scissors that can cut the DNA, but it doesn't happen randomly. The Cas needs a guide. He needs to know where to go, which gene and which site of the gene, and for this there is a guide RNA. And the guide RNA is something that we can produce in the lab, we can design very easily, and by that tell the CRISPR where to go and where to cut. So this is an example how CRISPR on the left sees the DNA that it was directed to and creates a break in the gene, and by that is doing gene knockout. Gene knockout meaning inactivation of a gene, hopefully a faulty gene if this is what we want, and there's no functional protein, and by that eliminate something that causes a disease. So what can CRISPR do? So it can do knockout, as I just mentioned, but it can also do a knock-in, meaning if there's a break in the DNA, you can now insert the gene that you want, the correct gene, and by that you can precisely correct a gene that is faulty. Not only that, you can use it as a tool to make genetically engineered mice, as we did in the GDRF. Also, CRISPR can interfere with genes without disrupting them, without causing any physical damage to the DNA. This is called dead CRISPR. It is dead, it cannot cut, but it can inhibit the gene. If it inhibits, there's also a possibility to activate the gene, to increase the expression of the gene. And I will discuss it now. How CRISPR can help us for GRIN disorders. So there are several ways we can think of, and this is one example. So here is an example of a normal gene, in this case GRIN1. And this is a GRIN1 that was mutated. There was a mistake, and instead of a G, it turns out to be an A. We can now design a guide that will bring the CRISPR to the mutated gene, but we don't want to harm the normal gene. We just want to knock out the mutated one. So the guide here will bring the Cas, but this Cas won't bind because we engineered it to recognize a specific place in the GRIN1 gene that appears only in the variant, but not in the normal gene. So there's no binding and no activity of the CRISPR. But here in the variant, because of the A, now the CRISPR can bind and cut, and by that causing knockout of the gene. This is the way we only abolish the activity of the faulty variant. We can do something else. We can do editing, and we can do it with the help of the base editor, which is another new technique in genetics. Again, this is a GRIN one, a normal allele, and a mutated allele where the G became an A and caused a disease. Again, we design the guide for the CRISPR, and the Cas cannot bind and goes away. Here, now because there is an A, the CRISPR can bind, but now it's a dead Cas. It doesn't cut. Instead, it recruits another enzyme, another protein, which is a base editor. And what it is able to do is to change the A back to a G. And by that, actually, we reverse the situation, and we have now two normal genes. This is called editing. And the last example how CRISPR can help us is the technology of CRISPR-A called CRISPR activation. This is suitable in cases when we have a loss of function mutation, when we have only one copy, and therefore we have half the amount of the protein that we want. So it is called a haploinsufficiency. In this case, CRISPR activation can be used as a tool to upregulate expression of the normal copy, to make up for the missing second copy. So it can be used to upregulate a gene or to replace a gene that was deleted. You just boost the normal gene to act more and to give eventually enough protein for a normal function of the normal function and preventing a disease. I would like to take a different example of a disease that causes obesity, not something about brain disorders. So in this example, there is an important gene that is responsible for regulation body weight. When it doesn't function properly, when there is less amount of it, then the animal or the patient will eat a lot and gain weight and wouldn't be able to lose. What we can do is to boost the normal copy that is left by attaching the CRISPR-Cas to an activator, and the one gene that is functional will have twice the activity, and the phenotype, the mouse, will look normal. So this technique was published this year in Science magazine, very important scientific newspaper, by a laboratory from UCSF. And we can see here the situation when the CRISPR activation causes the gene to work harder, the normal gene that is functional, to work harder. You can see here on the left the mouse who has the genetic disease that causes obesity. And then he was treated at the age of a few weeks with one treatment of CRISPR-A. And for that, he became a heterozygous CRISPR-A, and he looks just like a normal mouse. He's not obese, and he has a normal life expectancy. And this one treatment lasts for his entire life. So this is a technique that has a huge potential to treat diseases that lack gene function. It doesn't cause a genetic change. It doesn't change the genetic material. They just make the one gene that is active to work harder and to compensate for the mutated gene. I would like to quote Dan Rather, a very famous journalist, who spoke about the CRISPR revolution and said, "This has the potential to be bigger and more important than the internet." Gene editing is one of the most important scientific and political tools of our time. The potential for improving public health is immense, but so are the potential risks. So we know about the case of the Chinese scientist that wanted to improve normal human beings, make them resistant to HIV infection. But this is not the case that we are talking about. We don't want superhumans. We just want them to be disease-free and cure them. CRISPR has many applications in medicine. Drug discovery is long, laborious, and expensive, and CRISPR can help us accelerate the preclinical stage of developing new drugs. There are CRISPR screening libraries that can facilitate discovery of new drug targets. Also, CRISPR can be used for creating disease models to validate potential drugs. We in the GDRF already use this technique to produce mice carrying human mutations as models, and it took us only a few months in a process that usually took several years in the past. Also, we want to use CRISPR as therapy, as a genetic manipulating tool. It will enable development of better therapies and personalized medicine in the near future. And this is not something that will happen in a long time, because there are already clinical trials with CRISPR. I looked at the clinicaltrials.gov, where all the clinical trials in the world are represented, and there are already clinical trials with CRISPR. For instance, for genetic disease of anemia, HIV infection, and also malignancies, and there are many to come. So I would like to summarize. I didn't discuss drug therapy in this webinar. I just focused on gene therapy and mentioned several strategies to treat genetic disease. One was gene replacement therapy, good for deletion or nonsense. The other was gene editing for missense mutation, and gene activation when we want to increase production of a normal protein. Now, research takes time, takes a lot of resources and motivation, but we have the technology here, it exists, and it's rapidly evolving. And I think that we have a responsibility as parents to look for a better future for our children, not only helping them with daily activities, but to make sure that they will improve their potential, eventually be independent. This is something that is very critical, and we can really think about cure, not just hoping and dreaming about it. Thank you.